everyone. My name is Indrani Sasmal. I am the grant writer at the college. Well, I did my PhD from South Dakota State University. My profession is a wildlife biologist, so I did my PhD in wildlife biology from South Dakota State University. And while working on my PhD, I did some DNA extraction of foxes. So I did some molecular genetics there. Then I did a postdoc at North Carolina State University. Then I came to join Dakota College at Bodno because of how great the location is, the community, and the college has a lot of potential. They have very good programs. They have very good instructors, very good professors who are always ready to help students. So you can come and explore DCB anytime you want and see how good great programs they have. And it's like not that many students, so there is always one-on-one interaction with the professor. They develop great relationship with the students, and the students end up in great places. So it's a small place, but you can go to great extent from here. So today I'm going to demonstrate how you extract your own DNA. So when I say your own DNA, well, in a short span of time and with the limited resources, we cannot expect to extract pure DNA. So there might be other DNA like that is inside our body, like microorganisms, many microorganisms like bacteria, whose DNA will also be extracted along with our DNA. We need many more resources to get pure DNA. But what we are going to extract today will definitely have our own DNA. For that, we'll need some tap water or drinking water, salt, just like plain salt, not the iodized one, some dishwasher detergent. If it is colored, then it's better, and non-colored detergent will also work, and some isopropyl alcohol or rubbing alcohol, and clear cups. So what we are going to do today is dissolve some salt in drinking water. So here in cup number 1, what I did is take half a cup of water, around 6 ounces of water, and then dissolve 1 tablespoon of salt, like normal kitchen salt, in that water, and make sure it's dissolved all the way. I mean, there is nothing left, residue left in the water. Then in cup number 2, what we did is mix 3 tablespoons of water with 1 tablespoon of dish detergent. And we take a third cup, which is an empty cup. You can take out the rubbing alcohol in another cup, or that would make it easier to pour it from the cup. So, and place it aside. Now this is a common solution that we are going to use to extract DNA. So this can be used by multiple students. It can be a stock solution. We can do the same for the soap water as well. Like each student can have their own soap water, or there can be a common stock kind of soap water where you can take 1 scoop out or 1 tablespoon out. So why are we using salt water? So what salt does is it helps loosen. So what we will do is we will take the salt water inside our mouth and spit it out. So what salt is doing there is helping loosen our cells inside our mouth, and it also helps in getting this water out of the cells so that the cell can stick together. Now, what dish detergent do? Why do we need dish detergent? Is what dish detergent do? It helps breaking up the cell membrane so that we can get whatever is inside the cell. So generally, DNA we found inside the nucleus of the cell. So we need to break the cell membrane, we need to break the nuclear membrane to get to the DNA. So that is what the soap water or the soap dish soap is doing. Just to not waste resources and reuse as much as possible, taking care of the environment, what we can do is we can make a stock of salt water with dissolving like 1 tablespoon of salt in half cup or 6 ounces of water, and then take 1 teaspoon of this salt water in a smaller cup or glass and give it to each student. So each student can use this water. Same thing we can do with the soap water as well. Take it out in another small cup. So for 1 teaspoon of salt water, we will need 1 tablespoon of soap water. So now what I will do is I will take 1 teaspoon of salt water that I prepared from the cup and put it in my mouth. I know it's very salty, it will taste horrible, terrible, whatever you can say. You can drink water after that. I know, it looks gross. Tastes gross. So, okay. So this is now your spit, which has thousands, millions and billions of DNA, I should say. Not only your DNA, but also the microorganisms, the bacteria that is inside your body, that DNA as well. Now what we will do is we will take 1 tablespoon of soap water and mix it with your spit. So do this gently. Do not take a stirring spoon, stirring rod, whatever, and mix it gently. Do not like shake it, or I don't know what will happen, but that might not work. So okay, mixing, and so keep it in your mouth. Try to for 30 seconds or so, work it in your mouth so that you get all the cells, as much cell as you can from your mouth. And try not to eat right away, right before doing this experiment. Then you'll have food also in there, so that will be more gross. So mix it. So once it's mixed, what we will do is take the rubbing alcohol, and we will put 3 tablespoons of rubbing alcohol very slowly or gently. Try not to mix the rubbing alcohol with the solution so that it floats on the top of the water, or your spit and the soap water solution. Now DNA is very, very helpful. Every individual have their own unique DNA, except maybe identical twins they might have, and that can be helpful in many branches of study, like forensic study. If we want to do, like for my PhD, like wildlife studies, also we used DNA. DNA can be used in many fields of studies, scientific studies. So molecular biology or molecular genetics is kind of a branch of study which can be used in animal sciences and plant sciences and environmental sciences and all sorts of fields. So we keep this aside for a minute and for the DNA to clump together so that we can see them isolated at the bottom of the alcohol, like where the alcohol separates from the solution. So this experiment I already did earlier, so you can see how the clouds of DNA. And so longer we leave it, we can see the more DNA kind of like accumulates. See, this one, if we compare, it's just started getting the bubbles. So the bubbles, the DNA are associated with the bubbles. It's coming out, it's separating. It will take some time to separate. And this one, while it has been sitting there for around 5 minutes, you can see the difference between these two. Like it has got a lot more DNA than this one. I love molecular genetics. I love how DNA works, how, like, even if there is a murder, like forensic sciences, there is a murder and you don't know who that is, you can, even from a strand of hair, you can isolate that DNA and find out that person who it belongs to. So, like for plants, we can genetically engineer plants which are more resistant to the weather, like drought or cold, so that we can grow that variety of plant or resistant to disease. So, like for my study, what we did is we were trying to find out how diverse the population is, and the more diverse the population is, the better its chances of survival. So that's how we apply DNA or molecular genetics in wildlife studies. There are many ways or many aspects of molecular genetic studies. You can go. I'm fascinated by genetics. So, yep, we can see how clouds of DNA are accumulating here. I don't know how good you can see, but once you do it, you can definitely see it. Yep. This would look better. There are tons of DNA. I'm sure you'll get it, and you'll get very excited if you have electron microscope. So these are DNA clumped together. This is not single strand of DNA. So if we need to do electrophoresis to isolate the DNA, and DNA are made up of deoxyribonucleic acid, and it's unique for every individual. Thank you.